Hello, and this is part one of a three-part lecture series on viruses, viroids, and prions. So we'll be looking at this very relevant topic in today's COVID world and discussing characteristics mainly of viruses. Most of the entire chapter is devoted to viruses, and we'll talk a little bit about viroids and some about prions as well. Prions are interesting little protein particles that are similar to viruses but not, and the same with viroids as well. So let's get started. So some characteristics, basic viral characteristics. They contain DNA or RNA, but never both. So you have, and this is one way that we will classify them. This is kind of like the main classification division. Is it a virus that contains DNA, or is it one that contains RNA? And if it contains RNA, by the way, we call them retroviruses. And the reason why it's called retro means in reverse, because they start with the RNA and they use then the RNA to develop through a series of processes DNA. And under what we would say, quote, normal cell circumstances, you start with the DNA, and from the DNA you get the RNA, but they go in reverse or retro, so we call them retroviruses. They all possess a protein coat that surrounds their nucleic acid, and they come in a variety of shapes depending on what type of virus family it's in. Some are enclosed by an envelope, and that envelope is usually a lipid envelope with some other bells and whistles, some other molecules that are associated, but primarily it's going to be a lipid envelope that surrounds it, almost entirely a lipid envelope. And if you remember from the last chapter about the least resistant microbe, I guess you could say, or viruses technically aren't living things, but we'll call them a microbe. Those were the least resistant of all of the microbes. If a virus with a lipid envelope. Some viruses have superficial spikes that serve as a means of attachment to their host cell, and these spikes all stick off their surfaces. Influenza virus is notorious for its spikes. Coronavirus has spikes all over its surface as well. So a number of them do. And these spikes are kind of like, imagine them being like a key that goes into a lock on the host cell surface, on the host cell membrane. And then that key goes into the lock, the cell welcomes the virus in, and then the virus can do a number of things depending on what type it is, but basically overtaking, hijacking and hacking the nucleic acids that are in the host cell in order to make more of themselves in some way. Most viruses infect only specific types of cells in one host, so there's a high degree of specificity with viruses. Typically there are. When viruses when they evolve the ability to infect other species, that is a sign of alarm. So they develop these adaptations, their genes will shift and they will mutate, and in some cases they're able to enter other species cells or even groups of animals. For example, influenza being an entirely avian flu originally is able to infect pigs or swine, and of course humans as well. But originally it is theorized that influenza was exclusively an avian or a bird virus, which has been able to adapt its way and move its way into other groups of animals. All vertebrates going from bird to mammals, but it was able to do that through what's called antigenic shift and antigenic drift, or genetic shift, genetic drift. And we'll talk about that later in this chapter. Host range is determined by specific host attachment sites, and that's the key. There need to be specific attachment sites for those spikes to be able to attach themselves to. And then through what's called a type of endocytosis, similar to receptor-mediated endocytosis, where the cell basically takes the virus in because the keys go into the lock, the virus then is taken into the cell. It was first isolated and studied in 1935, the tobacco mosaic virus. So that's when it was first understood that there was this viral particle, and they didn't quite know what it was. So they called them viruses, which comes from the Latin word which means poison. And so they thought it was some kind of a poisonous substance, didn't know what it was. The tobacco mosaic virus, it was first begun. Its study in the late 1800s, I think like 18 in the 1890s, somewhere around there, 1894, 1896. But it wasn't until a few decades later in 1935 that we could determine what they were. And it was in this virus that afflicts and devastates the tobacco plant. I'm going to talk a little bit about these next two things for a few minutes, so sit back and relax and pay attention. So phage therapy versus antibiotics. So there is this, you need a little bit of history. So United States and Russia at the beginning, around the beginning of the 1900s, around we'll say 1905, 1910, somewhere in that ballpark, both nations were researching and studying and working with this therapy called phage therapy. And what phage therapy is a phage its full name is a bacteriophage, and it's a virus. And these are viruses that only can infect prokaryotic cells, bacteria. They cannot enter eukaryotic cells because the cell wall, cell membrane, biomechanics and biology of eukaryotic compared to prokaryotic are so vastly different. And so there are these, and there are literally almost an uncountable number of phages on Earth. In fact, they could be considered, I guess, technically the deadliest thing on Earth because they kill bacteria by the bajillions every single day across the globe. So there's a good video by Kurgaset, I think is how you pronounce that, about phages. And it's a bit sensationalized, as a lot of Kurgaset videos are, but it gives a good basic introduction to what a phage is. But anyway, United States and Russia researching these viruses that hunt down, destroy bacteria. Okay, so we're doing research and study on this. Then, 1928, Sir Alexander Fleming, a Scottish physician, he discovers antibiotics. So he's doing research in his lab. He notices that mold growing on his petri plates that were growing bacteria, that the mold inhibited the growth of the bacteria. And so he surmised that the mold must be producing a chemical compound that is killing the bacteria. So imagine a petri plate with some fuzzy mold growing on it, along with bacteria all around the mold, except for a ring all the way around the mold growth. And so what he deduced was that the mold was producing a compound that was kind of like diffusing out of the mold body, the thallus, and was going into the surrounding medium. And anytime the bacteria encountered this chemical compound that was produced by the mold, the bacteria died. They couldn't survive in it. And so that was the discovery of antibiotics, and it was an accidental discovery as well. But the genius of Alexander Fleming was that he noticed it, he researched with it, he explained it, described it, and so forth. So he discovered it in the mold penicillium. So the first antibiotic that was developed was penicillin. This was in 1928, and Alexander Fleming's from Scotland, the United Kingdom. The United Kingdom and the United States are allied nations, they're friendly nations, and so as a result of that, the United States abandoned phage therapy. The Russians did not because they were the, quote, enemy at the time, and so they did not jump on board with this antibiotic research. Instead, they remained with phage therapy. So, United States, Great Britain, and other developed nations of the world went with antibiotic research and therapy around especially the 1950s, after World War II. And while Russia, the USSR, now Russia again, continued researching phage therapy. Now, why do I talk about this? Well, with antibiotics, as you know, there is a global problem with multi-drug resistant bacteria. These bacteria have developed, they've adapted, they've evolved their way around the antibiotics that we are giving them, so that the antibiotics that were administered 50 years ago are virtually useless today. And so we have to keep developing new classes of antibiotics, new and more powerful antibiotics, to the point where we have to be very careful because we're going to develop antibiotics that are going to be harmful to our cells as well. Notice they're antibiotics, anti-life. And so the bacteria, because they reproduce exponentially, and so they can undergo adaptations at an alarmingly fast rate, they are working their way around our antibiotics, becoming resistant to the antibiotics, and so they scoff at our antibiotics now. And we're running out of options. And to the point where, and we're seeing this more and more, where they're taking, say, for example, the antibiotic that's locked up in the vault. We don't want to use this antibiotic. We want to save it as a last resort. Absolutely do not administer these antibiotics unless it is a life or death situation. Those antibiotics are now having to come out of the vault more and more because the bacteria are developing so much resistance against our antibiotics. And if we continue at the pace that we're on, with the bacteria evolving their way around antibiotics, and us taking years and years, 15 years, to develop new classes of antibiotics, we're going to run out. And it's going to be like a pre-antibiotic world, before there were any antibiotics to combat any bacterial infection. And so then you're just left with your immune system, and that's it. To ward off the infection. Hopefully you have a strong immune system, because there aren't going to be any antibiotics available to us in a few decades if we continue on at the pace that we are. Now, of course, with human ingenuity and development, there's going to be some research and development, obviously, over the next few decades. So I'm not preaching Armageddon here. It's not the end of the world. But we're eventually going to get to the point where we're in an emergency situation. Well, guess what? Phage therapy is still being developed and researched by Russia. What phage therapy does, and what's interesting about it, is that phages are, remember, they are viruses, and as such they have nucleic acids. And so they can be genetically engineered and modified by humans, or they too will adapt to, like if bacteria evolve resistance against these phages, the phages will just evolve right along with the bacteria. So it's not like an antibiotic, which is a stagnant chemical compound that has no ability to do any kind of genetic shift or drift or anything, because it has no nucleic acids in it. Phages can. So as the bacteria develop resistance against the phage just develops anti-resistance against the bacteria. And phages are viruses, so they're highly specific, like all viruses are. So they will only target a specific species. So let's say you have some kind of a strep infection. You can undergo phage therapy. In other words, you're going to have phages injected into you, or they're developing pills now to avoid the use of needles and risk of contamination and cross-contamination and all that kind of stuff that are associated with needles, where you can take a pill, it'll get absorbed into your bloodstream, those phages will be released, and they will only attach to those strep bacteria. They will not wipe out all of the bacteria that are in us, which is what antibiotics do. Antibiotics are completely non-selective. Well, I shouldn't say completely, but they are very non-selective. If it's a bacterial cell, they kill it. Phage therapy does not function that way. If it's a particular strain, or whatever the strain happens to be of strep, that's the target and nothing else. It won't kill the E. coli, and it won't kill all the many other normal microbiota bacterial species that we have in us. So phage therapy, highly specific. Antibiotics, very non-specific. And bacteria developing resistance against the antibiotics, but they don't develop resistance against the phages. And what's interesting is that, and they've done research on this, as the bacteria start to develop resistance against the phage, and it looks like the phage is losing and the bacteria is winning, what they found out was that, okay, let's take these old antibiotics and see what happens. When they administer the old antibiotics that used to be really good, now they're useless because of bacterial resistance. When they administer those old antibiotics to these bacteria that are developing resistance against the phages, the antibiotics kill them. So the bacteria seem to be in what we could say is like a catch-22. As they develop resistance against antibiotics, the phages nail them, wipe them out. As they begin to develop resistance against phages, the antibiotics will nail them and wipe them out. So sometimes you might see articles or maybe the media say, well, this antibiotic resistance, we're not going to have anybody left on this globe in 20 years because of this antibiotic resistance in bacteria. We have phages. Actually, the Russians have phages. The place that's doing the most amount of research is in the Republic of Georgia, which used to be one of the I don't know what you call them. I'm not a history person or a government, what do I say? Social studies person. The Republic, it used to be a Republic of the USSR. It's now just the Republic of Georgia, its own entity now. There's a Phage Therapy Institute, and you can Google them and find out more. So, very interesting, very effective alternative. And phage therapy has been around for over a century. Since the early 1900s, around 1910, in that ballpark, is when phage therapy was being first ushered into laboratories and being researched by scientists and at universities. So this is a great alternative to antibiotics. So watch for phage therapy to start gaining momentum. You're going to see it more and more as the government gives more and more money to this research. Phage therapy will become more available. Oncolytic virotherapy versus chemotherapy. Um, Chemotherapy has been the traditional treatment for cancer, of course. Well, the side effects of chemotherapy are awful. If you or anyone you know has undergone chemotherapy, it's devastating to that person, to the victim of the cancer. They become nauseous, they lose their hair, they get skin lesions, and among many other things. And the reason why is because, like antibiotics, non-selective, chemotherapeutic agents are non-selective as well. If it's a rapidly dividing cell, which cancer cells are, it gets nailed. So what are rapidly dividing cells? Your gut cells, your skin cells, hair cells. Rapidly dividing cells, they get nailed by the chemotherapeutic poison, really is what it is. It's poisoning cells. They get nailed, your hair falls out, you develop nausea, vomiting, and all the other nasty side effects that are associated with chemotherapy. Now, it works against the cancer cells, sure, but there are awful side effects that go along with it. Oncolytic virotherapy is a great alternative to chemotherapy. Chemotherapy we've been doing for decades, for a very long time, and chemotherapeutic agents have gotten better. We've diversified them, we've made them somewhat more specific, but still it creates devastating effects for the body. Oncolytic virotherapy, so look at the term. Onco means cancer, lytic means to split or to burst. So this is a type of virotherapy that's causing cancer cells to burst. It's also called oncolytic immunotherapy as well. So what's happening? These viruses are genetically modified so that they will target, hunt down, and destroy cancer cells. Cancer cells, when they become cancer cells, are very transformed cells. They're nothing like any other cell in your body, and so they become a very unique cell with unique markers all over their surface. Those viruses will target those cell membrane locks. Okay, so these markers are very specific to these cancer cells. These viruses, these oncolytic viruses, have been genetically modified to latch on to those markers, to those attachment sites, enter the cell, cause the cell to burst, release all kinds of cancer junk, cancer proteins and carbohydrates and so forth that get released into the bloodstream, and as a result they educate the immune system, and so now T cells get in on this. They quote figure out what these new antigens are that have been burst out of these cancer cells, because the cancer cells never got to the point to divide. Now their bits and fragments are circulating through the body rather than being contained within a tumor, very privately, just multiplying and dividing and overtaking an organ and leaching all of its nutrients in its blood. These fragments get released. They then get picked up by immune system cells. The immune system gets educated, and now your own immune system now can fight the cancer. And that's why it's called immunotherapy. There is so much more research that needs to be done in this oncolytic virotherapy. The Cancer Research Institute is doing a lot of therapy with this. There's a ton of money going into this research institute as they develop more oncolytic virotherapeutic agents, viruses, in order to combat cancer as a viable and safer and far fewer side effects than chemotherapy. Phage therapy, same thing, far fewer side effects. There's no risk of secondary infections with phage therapy as there is with antibiotics, because when they wipe out the normal flora, now you're at risk for C. diff or yeast infections and so on, secondary infections from taking antibiotics. With oncolytic virotherapy, it's a very specific target, and you don't have to receive a poison in order for these cancer cells to be wiped out and killed. So, a lot more I could say on that. It's a very exciting thing. Phage therapy is, and like I said, it's been around for over a century, but it's been so quiet because of antibiotics getting the push and all of the money and all of the research and all of the attention. Same with oncolytic virotherapy. Chemotherapy has been getting all the push, all the money, all the attention. And oncolytic virotherapy is like in the background. Hey guys, look over here. I'm a great alternative to chemotherapy if you would just pay attention to me. And I think we need to pay attention to it, to both of them, phage therapy and oncolytic virotherapy. All right, enough said. Moving on. So, are they alive? Well, viruses are inert outside a host cell. That makes them non-living. They can't do anything. So they're, in terms of like virotherapy, they're kind of like a drug. A drug is inert outside of a living body. You take the drug and it does something. Viruses are inert outside the body, but once they get into your body, they do something. So when you talk about viruses in terms of virotherapy, they're basically like a chemical drug that we take, a medicinal drug. But so they're non-living. They're not doing anything outside of the cell. They have few or no enzymes of their own, so they need the host cell's enzymes. They're obligate intracellular parasites. They have to be inside a cell in order to make more of themselves. However, once they enter a cell, their nucleic acids become activated and they start multiplying, just like any other living thing. So we could consider them a very complex mixture of chemicals or a very simple living organism. I'm leaning more towards the biology side of things, so I'm going to call them a very complex mixture of chemicals. However, you get them inside and they cause disease just like any other pathogen, so that makes me scratch my head a little bit and say, maybe, but no, I'm going to stick with them being non-living, very complex mixture of chemicals. Virion is a complete, fully developed infectious viral particle. It's got its nucleic acid and it has a protective protein coat that's called a capsid. So now we could say that it's like full grown, fully developed, ready to go out and cause infection. Enter another cell, make more of its cells, cause the cell to burst. The cell bursts, release all the baby viruses, and you get the idea, and it goes and spreads. A capsomer is a subunit of a capsid, and I only mention that because we use it to identify particular viruses. Capsomer. An envelope is a lipid, protein, and carb complex, mainly lipid, covering the capsule. Many times contains parts of host membrane in order to allow ease of entry into the host cell. And a spike is a carb protein complex that comes off of the envelope. It's like a key. And so that key matches a particular lock on a host cell, and then in it goes. We use spikes to identify the virus as well. So like for example, you know about the influenza virus, H1N1, H1N2, H1N3, and so on. Those H's and N's are spikes. And we identify each one of them differently. And by using these subclassifications of H1, H2, H3, and so on. So it attaches a virus to the host cell, and it also causes cells to clump together so that viruses can go, they like to agglutinate cells, so they can just pass from one cell to another, and they don't have to leave the cell, go into the extracellular fluid, be recognized by white blood cells or other immune cells, and then get targeted. So if they can clump cells, that makes a very quick, easy, efficient transfer from one cell to the next. This is just saying viruses are really, really, really small. Human red blood cell is not a very big cell at all, and it's enormous compared to these viruses. So, viruses are very small, and because of viruses, tobacco mosaic virus was first researched and studied in 1935. It coincided with the invention of the transmission electron microscope, and we needed that transmission electron microscope to actually see the virus because we had nothing powerful enough to magnify something so incredibly tiny. So interesting that those two were kind of parallel. Understanding, identifying tobacco mosaic virus And what was needed was a transmission electron microscope, which came along in 1935. So that was handy, and that's just they're so small. We have to use that level of microscopy. We're going to look at basic shapes of viruses. There are viruses that are helical, so they have a hollow cylindrical capsid that has a helical structure, and the nucleic acid is wound up inside of it, like the Ebola virus. Ebola is a helical virus. Second one is a polyhedral virus, where poly means many, hedral, sides, so it's a many-sided virus, and inside of it is a nucleic acid all wound up like a mastodinovirus, for example. And you can see the spikes that are projecting off of the corners of this virus. You see those kind of wispy white lines with little knobs at the end of them? Those are the spikes. And in those little knobs are enzymes. And those enzymes enable the virus to enter the cell. And they're used to break down parts of the cell membrane so that the virus can gain entry. And then there are complex viruses like bacteriophages, which look fake, science fiction almost, because of their unusual, almost unbelievable structure. They look like they're some kind of landing module landing on the moon or on Mars. They call them complex because they have complicated morphologies. They have additional structures besides what you saw in the helical and the polyhedral virus structure. What these guys do is they land on a bacterial cell, they inject their DNA into the bacterial cell, so he's going to just give himself up for the greater good, and from that one bacteriophage can come millions of new ones that will go and infect other bacterial cells. So again, I can't say enough about phage therapy. What happens is you just take, it's like for the administration of it, it's one and done. You take a pill, you get an injection of the bacteriophages, they will land on the bacteria, and then from that, they'll just self-replicate. And they'll just self-replicate. And you don't have to take any booster, anything. You don't have to take another pill because they are self-replicating viruses. And then once the bacteria that's causing the infection, once those bacteria are eliminated by the phages then will just die off. And they cause no ill effects. They can't infect eukaryotic cells. They cannot enter human cells. And so the body will just break them down, absorb them, consume them by white blood cells or whatever. And it'll be a totally harmless result from it. So anyway, sorry, I like to talk about that. How do viruses reinfect? So for example, why do we get the flu more than once? Well, as I mentioned, these surface proteins, the spikes, they can be altered due to genetic mutations. There's a whole lot of genetic shift and drift that are going on with these. So they can produce antibodies to one influenza virus. The virus can mutate and infect you again. The immune system and its relationship I'll get a little more specific. Antibodies and their relationship to antigens, which are non-self things like pollen or egg whites or peanut oils or the flu virus, they're highly, highly specific. So when you get infected by a particular strain of the influenza virus, you will produce antibodies specific to only that strain. So if a new strain infects you again later, it's like you're totally reinfected again. Even though it's the flu, it's a new strain, and in terms of your immune system, it's brand new. So you have to develop a whole new set of antibodies against that newly mutated strain of the flu. So we keep getting it again and again and again. But some viruses, they don't mutate nearly as rapidly as the flu virus does. And so you get infected once and then you don't get it again. Like, for example, Epstein-Barr. That's one where most people only get mono once. And they don't get it again because you develop antibodies against them. So this is one of the reasons why. There are other reasons as well, but this is one of the primary reasons. It's because these spikes, these carbohydrate protein complexes that are projecting from the surface of the virus, they are mutating. And what that means for your immune system is, got to make a whole new set of antibodies because it's like a brand new infection all over again. Okay, that's the end of part 1. Stay tuned for parts 2 and 3. They'll be coming up. I'll be recording them shortly. So again, thank you for watching. Hope this was a benefit to you. And stay tuned for part 2.